Good evening, everybody. My name is Eric, and I am the host of the Men Managing Myositis through TMA's website. Tonight, we are hosting Dr. Iago Pinal-Hernandez of the National Institute of Health down in Bethesda, Maryland, and he will be presenting us tonight. He is with the Muscle Disease Unit, and he has a presentation for us tonight for those interested in the myositis field and those suffering from it. And afterwards, after his presentation, he will take some question and answers from the group. So again, just so everybody knows, this meeting is being recorded for the use of TMA. Dr. Pinal, welcome. Thank you for coming on tonight.
Thank you so much for inviting me. I'm really excited to share this with you. Today, I'm going to be talking about pathogenic autoantibody internalization in myositis. And I have to tell you, you are the first ones that are going to hear this after this has been publicly released. This was released last week, so it's super novel. We have talked about some of these things in some international meetings, but last week it has been made public, the whole study. And the reason why we made it public before it has been peer-reviewed is because we think that the sooner other groups start validating these findings, the better for the advance of the disease, the better for our advancing our knowledge about the myositis pathogenesis, and the sooner that we're going to start using this knowledge. The second reason is because we think this is important to understand the disease. Okay, so a little bit about my background. I'm a physician specializing in autoimmune systemic diseases, particularly in myositis. I work at the NIH. I'm also affiliated with Johns Hopkins with the Myositis Center. And at the NIH, we see most of our patients through these natural history studies, the natural history of inflammatory muscle diseases, led by Dr. Mamin here at the NIH. If any of you is interested in participating in this study, here is the contact information of our research nurse. You just send her an email that you're interested in participating, and she will be happy to coordinate the visit. I'll put this at the end in case anybody wants to get the email. And when we see patients here at the NIH, it's pretty much similar to a normal clinical visit. The only difference is that we get some biospecimens to do, extra biospecimens to do research. So, as you know, in myositis, there are different types of myositis, and this has been changing over the years, how we group them. The most low-level classification is based on syndromes, on collections of symptoms that patients have. Dermatomyositis is one of these groups where patients have muscle involvement and skin involvement. The antisynthetase syndrome is when you have one of the antisynthetase antibodies and some characteristic clinical features. Immune-mediated necrotizing myositis, mostly muscle involvement and the characteristic antibodies if they are present. Inclusion body myositis, and I have heard that most in this room suffer from this disease. I'm going to talk a little bit about that at the end. And overlap myositis, which doesn't mean that is confusing or it has things that we don't understand. No, patients with some types of overlap myositis, they have very specific phenotypes that are very, very characteristic. And the one that I'm going to focus today is the one that is induced by these antibodies and the antibodies. So one of the most relevant advances in our understanding of myositis has been the discovery of autoantibodies. So antibodies that bind things inside of the cell and that are associated with very characteristic phenotypes. So in dermatomyositis, the four most common antibodies, they are not the only ones, are anti-MI2, anti-NXP2, anti-TIP1 gamma, and anti-MDA5. In the antisynthetase syndrome, the three most common ones are anti-Jo1, PL7, PL12. Sometimes the name of the antibody is related to the protein that they target. Sometimes it's because of the first patient that was diagnosed with this antibody. In immune-mediated necrotizing myositis, anti-SRP and HMGCR. In overlap myositis, there are others, but I'm going to focus about the anti-PMSCL. And in inclusion body myositis, this antibody is present in about 60% of patients with inclusion body myositis, and it goes against NT5C1A. So in the study, we talked about other autoantibodies, but today I'm going to focus on these four. 3 of them, we have good data in this study. The other one, I'm going to show you something very preliminary that we have that is not in this paper. So first, I'm going to start with anti-MI2. This is not the most common antibody that we see in patients with dermatomyositis, but it has been extremely helpful to understand the disease. So the first clue that the antibodies were important in this disease is when we measure the amount of antibody in patients with these antibodies over time. So here in the x-axis, we followed patients between 5 and 10 years. And this in the y-axis, you have the amount of antibodies. So you see that all of these patients, each one of the lines is a different patient. All of them started with pretty high titers of the antibodies and then tended to go down. But the interesting thing were these 3 patients that went below the detection threshold. The interesting thing that of these 3 patients, 2 of them could stop medication and never relapsed. So this was the first clue that, hey, these antibodies may not only be a nice biomarker, but if you don't have them, the disease may stop being active. So they may be something else. The second thing that was striking to us when we looked at the muscle biopsies of these patients is that they had something that shouldn't be there. So this is a muscle biopsy. Muscle fibers are really long. So this is cut cross-sectionally. And each one of these pink purple circles is one muscle cell. And you can see that there are some cells that are white, right? So mostly here on the periphery. These are cells that are dying. And this, we call it necrosis. And we usually see it not in dermatomyositis. We see it in necrotizing myositis. But these patients, only the patients with dermatomyositis and anti-MI2, they have lots of necrosis. And you see that these cells that are dying are surrounded by healthy cells. And this was very, very puzzling to us at the beginning. So patients with anti-MI2 antibodies, they recognize this protein here, the CHD3, CHD4, and probably also the CHD5. And these proteins are part of this complex called the CHD4 NURT complex. What this does is in charge of making each type of cell in our body make what they should make. So make the muscles make muscle things, make the pancreas make pancreas things, make the blood cells make blood cells things. And this is going to be important later on. And this complex is in the nucleus of the cell. It's in this part of the cell where all our genetic information is stored. And this also is going to be important as we move on. So many years ago, I started doing this type of analysis called RNA sequencing. And it was thanks to the support of the TMA. With this technique, what we do is we smash the muscle biopsy, we extract the RNA, which is what the DNA is translated on to make proteins. And we do a library, we sequence it. And what we get from this is a snapshot of what the muscle is doing at any given point in time. So we get massive amounts of information. We get about 60,000 individual measurements from a really tiny piece of muscle, as small as you can see. And for this particular study, we use muscle biopsies from 668 patients, including about 40 healthy muscle biopsies. This is by far the largest study of this kind in myositis. And when I say by far, it's orders of magnitude larger than the previous ones. And when we did this study in patients with myositis and dermatomyositis and anti-MI2 antibodies, this is what we saw. So here, the different groups, NT is normal muscle biopsies, so healthy normal muscle from volunteers. The second one is anti-MI2 dermatomyositis. Third one is dermatomyositis that are not MI2. The next one is antisynthetase syndrome, IMNM, immune-mediated necrotizing myositis, and the last one is inclusion body myositis. So these were the top genes that we identified that were characteristic for anti-MI2. And you see that the signal is extremely strong. This SCRT1 gene is only present in MI2 patients. In the y-axis, you have the expression, the amount of RNA that we are seeing in these muscle biopsies. And it's a proxy for the amount of protein that we have. So SCRT is only expressing MI2, P2RX2 only expressing MI2. All of these genes are only expressing MI2. And these are just the top 9. There were more than 100 genes like these ones. And this is really dramatic. This is something that we usually don't see in this type of analysis. These genes, they were usually not expressed at detectable levels in non-anti-MI2 muscle biopsies or normal skeletal muscle. They were all high, they were all overexpressed, and they were not related to the interferon pathway, which is one pathway that is known to be important for dermatomyositis. Some of them were so bizarre that, for example, one of the ones that we didn't include in the study because it was borderline, but insulin was being expressed in the muscle of patients with MI2, insulin that usually is expressed in the pancreas. So we identified that there were more than 100 genes, and the expression of these genes was mutually correlated. So if one was up, the others were up. They were correlated with the titers of antibodies. So if you had more antibody titers, if you had more antibodies, you had more of these genes. And they were associated with disease activity. So if your disease was more severe, you also had more of these genes. But we wonder, what is causing this? So the first clue was from analyzing the set of genes that we identified. And when we compared them with animal models that had knocked down this complex, the CHD4 NURT complex, we found that they were the same set of genes. So they were probably related to this set of genes was probably related to the dysfunction of this complex. But how is this possible? So you remember that I told you that this is in the nucleus of the cell. So this CHD4, the protein that the antibodies bind, is in the nucleus of the cell. And in autoimmunity, there is still a dogma that antibodies cannot go from outside of the cell to inside of the cell. Just to exemplify this, when you look for antibodies crossing the cell membrane, and this is from Quora, this is not scientific literature, why can't antibodies cross the cell membrane? And they explain you a lot of things about why this may be the case. And this was a dogma. The antibodies shouldn't get into living cells. But when you go back to the 60s, 70s, 80s, there were some scientists that challenged this dogma. In lupus, Reichling in Oklahoma found that there were some antibodies in lupus that got into living cells. The other group that worked a lot on this was a Mexican group led by Dr. Alarcón Segovia. And I am not responsible for the choice of words for this title. This is totally up to him. But he studied in lupus that the antibodies were getting into live cells. And the thing with this line of research that was lost, nobody continued doing it, nobody continued developing this line of research, and nobody looked into muscle. So we looked into muscle. We stained for immune globulin. We looked where the immune globulin was located in the muscle of patients with anti-MI2 dermatomyositis. And here in blue, you have the nuclei. This is a stain that we use to look at where the nuclei are located. And in green, you have the immune globulin, and you see that here, all the blue dots are also green. In this cell here, all the dots marked with the arrows. Here on the periphery of this cell, also on this cell, and also on this cell. And this cell in the middle, you don't see that. You only see the blue without the green. So this means that the antibodies have gone through the membrane, and they have been accumulating little by little in the nuclei of the cells. This was a dogma that this couldn't happen. This is happening in this particular disease. This is another example of another patient, another biopsy, and you see in blue the nuclei, in green where the immune globulin is being deposited, and you see that the immune globulin is being deposited in the nuclei of these cells. So then we wondered, how can we confirm that the antibodies are causing the effects that we are seeing in the human muscle, in the muscle from our patients? And we wonder, hey, we are really good at culturing human muscle cells. We can grow them in a petri dish. And how can we get the antibodies in? Some of us have some experience with molecular biology, and we knew that to internalize DNA into cells, we could use electricity. So we wonder, hey, maybe we can also internalize these huge proteins inside of the cells using electricity. And we did that. So if you zap cells with electricity, you make holes in the membrane. And anything that is outside of the cell gets inside of the cell. So we put the antibody of our patients into normal cells, normal muscle cells, and then we did the sequencing. We looked at what was happening on those cells when we did that. And here you see different columns. Each column is a different sera from a different patient or a different individual. This is control sera from healthy volunteers. These are the MI2 patients. So these are 5 different MI2 patients. These are NXP2 patients, another antibody from dermatomyositis, TIP1 gamma, MD5, Jo1, HMGCR. We did it for all of them. And here in red is the expression of these proteins. And these are the same proteins that I showed you in the muscle of our patients. So if we get the antibody of our patients, we put it into normal muscle cells that don't have any disease, we get exactly the same thing that we saw in our patients. We get things that should only happen in the pancreas. We get things that should only happen in blood cells, in muscle cells. So our conclusion of this data was that the antibody gets into the nuclei of the cells in this case. It starts de-repressing things that shouldn't be made in muscle. And this is probably mediating the disease. This is probably causing what we understand as MI2 dermatomyositis. So now let's go to the second one, PMSCL. And this, we have shown for the first time this, we discovered this a few months ago. We discovered this a few hours before we presented it in ACR. Again, because we think that this is important. We think that if we discover it, everybody should be on the same page, because this can make a cure happen sooner. So in PMSCL, patients with anti-PMSCL antibodies, PMSCL stands for polymyositis scleroderma. Patients with this disease, they have muscle involvement, but they also have features of another autoimmune disease called the scleroderma. So their skin gets a little bit fibrotic. Patients with these antibodies, the antibodies recognize these 2 proteins, exos 9 and exos 10. And these proteins are in a complex called the RNA exosome, the nuclear RNA exosome complex. This is inside of the cell in the nucleolus, and this is in charge of degrading RNA that is not useful for the cell. So this is like the garbage collector of RNA for the cell. And you remember before I told you that MI2 was in the nuclei, this one is in a part of the nuclei called the nucleolus, a really small, tiny part of the nuclei that is in charge of cleaning up RNAs that are not useful anymore. So with our more than 600 biopsies, we did the RNA sequencing, we analyzed them, and these are the top genes that we identified in PMSCL. PMSCL would be this one here. This would be, again, normal muscle, MI2, NXP2, TIP1 gamma, patients with these antibodies, patients with anti-MDA5, other dermatomyositis that we don't know what antibody they have. We couldn't identify any of the known ones. Anti-Jo1, anti-synthetase patients without Jo1, HMGCR, SRP. And here we included also other myositis that are not, do not have any of these antibodies, and then genetic myopathies. And this set of genes only appears on PMSCL. And you can see the name of the genes. It's kind of a weird name, right? So this ENSG, this means that these genes don't have a name because they shouldn't be in the muscle. These genes, they don't have a name because they are not interesting for anybody, because they are not present in most cells in the body. The other type of genes that appear that is not in this top genes that I'm showing you are divergent transcripts, that are genes that are normally, that have the normal sequence, but they are in the opposite direction. And these genes without a name, these genes that are in the opposite direction, should be metabolized by this exosome complex that I showed you, that these antibodies recognize. So we did the same. We used electricity to get these antibodies inside of the cell. And in this case, these are the 2 anti-PMSCL patients that we use for internalization of the antibody. So we use the antibody of 2 different patients, and we put them in normal cells. And you see all of these genes without a name and these DT, divergent transcripts, they started popping up. This didn't happen in any of the other types of myositis when we internalize any of the other antibodies. And again, we stained for immune globulin. We looked where was the immune globulin deposited in muscle biopsies from patients with this disease. And you see here, then again, the blue is the nuclei of the cell, and the green is the immune globulin. You see small tiny dots inside of the nuclei of this cell. I'm gonna zoom in this one. I'm gonna show you a higher magnification image of these nuclei down here. And you can see that there is a small bubble inside of the nuclei. This is the nucleoli. So the antibodies in these patients are depositing inside of the nucleoli, not in the nucleus, as a structure that is smaller than the nucleus. And probably what they are doing is affecting the RNA exosome complex. So what we think is going on in this particular type of myositis is that the antibody is getting to the nucleolus, is inducing a dysfunction of the exosome complex. And all of these RNAs that shouldn't exist are accumulating in the cell. This is toxic to the cell, and this causes the disease. Now we're going to a different type of myositis. I told you about dermatomyositis. I told you about the overlap myositis. In the paper, we also have data about the antisynthetase syndrome. But now I'm going to tell you about HMGCR. So patients with anti-HMGCR antibodies are patients that have a really intense muscle involvement. And this was described by my boss here at the NIH, Dr. Mamen, and it was described in patients that had the myositis after taking statins. And what the reason why this association is interesting is because the antigen, the protein that these antibodies recognize, is the same protein that the statins block. This is called HMGCR. This is the cholesterol pathway. This is what our cells need to make cholesterol. And if you block these, you cannot make cholesterol. The interesting thing that up here is this acetylcoenzyme A, and this makes fat. This makes triglycerides. So we said, okay, this is not going to cause anything on the RNA because this is not the function of this protein. But if it failed, if this didn't work, this should accumulate, and we should see an increase in the amount of fat. This antibody, the protein that this antibody recognizes is not in the nuclei, is not in the nucleolus. This is in the smooth endoplasmic reticulum and the cytoplasm. It's outside of the nuclei, okay? So when we stained these patients for immune globulin, what we saw was that the immune globulin was being deposited outside of the nuclei. This is the blue, the nuclei, and you see that in this case, total opposite of what you were seeing before. This is black, this is not green. The outside of the nuclei is green, okay? So then we looked at fat accumulation in these patients, and we can stain fat with a staining called Nile red. And you see these red dots. This is not normal. This is extremely abnormal. There is a huge accumulation of fat inside of this of the muscle of these patients. We tested a large cohort of patients for these fat accumulations, and 90% of HMGCR patients had accumulation of fat in the muscle, compared to about 8% of all the other types of myositis that we tested. Here is the immune globulin. Again, you can see that there is antibody inside of the cytoplasm, but this was the striking thing. So what we think is going on is that the HMGCR is not functioning properly because of the antibody, and this accumulates the building blocks to make fat, and these cells are making a lot of fat. We don't know if the lack of cholesterol may be also causing the cells to die, but at least this is the evidence that we got related to the antibody causing the disease. And now I'm going to tell you a little bit about inclusion body myositis. Inclusion body myositis was not the main focus of the study. The reason why is because these antibodies, anti-NT5C1A, do not only appear in inclusion body myositis. They appear in 60% of patients with inclusion body myositis, but they can also appear in other groups of patients with myositis, dermatomyositis, etc. These antibodies are against a protein that is in the cytoplasm of the cell. What is interesting is that this protein is not present in all the muscle cells. It's only present in what we call type 2 fibers, only a subset of the fibers in the cell. And I'm only going to show you a couple of slides, one slide about this, and this is extremely preliminary. We are looking into it really hard, but I wanted you to know it first. So when we stain for immune globulin, you see that all of these cells are green. They have green inside of the cell. And these, I can tell you that these are the nuclei, and the nucleus were excluded. I can tell you that these looked like type 2 fibers, and when we tested if they were type 2 fibers or not, they were type 2 fibers. We also stained these biopsies for NT5C1A, looking at the protein, and these were the fibers that had the NT5C1A. So this is extremely preliminary data. We still have a lot of work to do on this, but this is a clue that the antibody may be more important than what we thought for the pathogenesis of IBM. So in conclusion of all the things that I was showing you, the internalization of antibodies seem to be cause a dysfunction of the autoantigen, and this may be the first trigger that causes the initiation of myositis. And now, why is this important? Why is it important for you if the antibody causes the disease? So for several reasons. The first one is because we have lots of antibody-lowering therapies that we can use at this moment, and we have been combining them empirically before we knew that this was the mechanism of the disease, or before we had evidence about this being implicated in the pathogenesis of the disease. Immune globulin. We use immune globulin, and immune globulin has many different ways that it can do its effect. One of them is by decreasing the half-life of the antibodies that your body is producing. A new medication that is being tested at this moment in myositis is called efgartigimod, and there are other medications that are of the same family. This medication contains part of the immune globulin molecule, the tail of the immune globulin, the Fc, and what it does is that it decreases the antibody half-life. It doesn't allow the antibodies to be recycled from the inside of the cell, and it shortens its lifespan. And these, if the antibodies are important, these may have a therapeutic role in myositis. Again, this is being tested. Rituxan. The large clinical trials with Rituxan in myositis, they didn't show to be effective, but they were clamping many different types of myositis together. We use it all the time, especially in patients with HMGCR and SRP myositis, in combination with IVIG many times. And in combination with IVIG, for some patients, this is really effective. And what the Rituxan does is it decreases the production of antibodies. It targets the cells that produce the antibodies, the precursors of the cells that produce the antibodies, and the antibodies, after a couple of months, they decrease. Then there are also anti-CD19 drugs that are available, and they will be tested in the next year, that will decrease the antibody production. They are CAR T-cell therapies. They are T-cells from the patient that are modified genetically to attack the cells that produce the antibodies. So this would be another way to decrease the antibodies. And they have been reported in 3-4 patients so far, but in these patients, they have been super effective. And then the next reason why that the antibodies are pathogenic may be important is because we can tailor therapies by understanding what is the mechanism of the disease. For example, JAK-STAT inhibitors. This is a group of medications that inhibit many things, but among them, they inhibit the interferon pathway. And we know that all the different dermatomyositis antibodies, they end up activating the interferon pathway. So JAK-STAT inhibitors is a medication that we are using more and more in patients with complicated manifestations of dermatomyositis. Dasukibart. This is a medication that is now undergoing clinical trials. The preliminary results of some of these trials have been very promising. And this is an antibody that blocks the interferon beta-1, which is the type of interferon that we think is pathogenic in dermatomyositis. And the other antibodies in dermatomyositis I didn't talk about, their function is basically suppressing or not inducing the interferon beta-1. So in dermatomyositis, again, if the antibodies are important, using these may be a really good option. And then mevalonolactone. Recently, it has been a new disease has been discovered, a new genetic disease that is caused by mutations in HMGCR. And in this type of genetic muscle disease, mevalonolactone was able to recover some of the function of the patients. And what this mevalonolactone does is that it can bypass the function of HMGCR. We are wondering if the antibodies are causing a dysfunction of HMGCR, if mevalonolactone could be effective as a treatment. Again, this is not even in clinical trials. This is in the planning stage. And the last potential application, and I think the most interesting one, if the presence of the pathogenic antibodies is the only and necessary and sufficient condition to develop each type of inflammatory myositis, it would be possible to develop curative therapies. Let me explain. So these are the cells that are producing the antibodies. We call it plasma cells. These are factories of antibodies that are releasing antibodies all the time at really high amounts. These are all the cells that lead to this plasma cell. And all of these cells, they have immune globulin in the surface. And we know what the immune globulin is recognizing. We know that it is recognizing the antigen. So one of the things that we have been working on here at the NIH, we have been engineering combining the immune globulin of the human immune globulin with the antigen of each one of the diseases to make a kind of a chimera of the tail of the immune globulin, which is what, if the antibody binds to any cell, can kill the cell. So this is called the FC region. And we have been putting the antigenic part of the antigen. In this case, for anti-HMGCR, the part that is recognized by the antibodies would be this green bubble here. So our final construct would have the tail of the immune globulin and the catalytic domain of the HMGCR. And this may have different effects. The first one is that it would bind to the antibody. So the antibody wouldn't bind to the HMGCR in the cell, and this may help to treat the disease. But this is not the most important one for us. The most important one for us would be that it could eliminate the cells that are producing the antibodies. I told you that these cells, all of these cells that are going to produce the antibodies, they have immune globulin in the surface. So this construct would bind to this antibody, and since this thing is toxic to the cells because it activates complement, it would destroy these cells. These plasma cells only live for a few months. So if we can destroy all of these precursor cells, we may be able to treat or even cure the disease. And this, again, this is something extremely preliminary. We have shown in vitro that we can neutralize the antibodies. We need to do animal models. We need to test it in humans. This is something that probably will take 5 to 10 years to develop. But it is something that if the antibodies are pathogenic, it is a possibility. And it is a possibility that we are working in really hard. So at this moment, we have developed similar drugs, not only for HMGCR, but for all the rest of the antigens, and we will be testing them in the next few years. So now I want to thank all the people that was involved in this study. This was not an NIH thing or an NIH and Hopkins thing. This was the contribution of many, many really nice people that worked really hard from many different countries. So at the NIH, Sandra Munoz-Braceras did many of the internalization experiments. All of our team here worked really hard on this particular project. In Spain, we had two really good, nice collaborators from the Clinic Hospital, the Valdebron Hospital. They were really instrumental for this. The Hopkins team, the Hopkins cohort were really necessary. And then we also got many of these biopsies from the Mayo Clinic, the Dr. Werner Stenzel in Germany. Dr. Steve Grimmer was really instrumental, not only to discovering the anti-5C1A antibodies, but also to help us understand some of the things that we were finding here. And now I'm going to leave you with the conclusion, with the information on how to be enrolled in our study. And now if you have any questions, I'll be happy to answer them.
Okay. Let's see, there's a.
I see a question in the chat.
Yep, we'll do those. I'll try and keep an eye on which questions are going in there. Some people might actually be raising their hands, like I see up on the screen here. I see Ricky and Colin both have their hands up. So let's take care of the chat one first. And the chat one is from Manuel Lubinas, and it's what is the mechanism mechanism of internalization for MI2 and other antibodies?
So that is a great question, and the short answer is that we don't know. At this moment, we don't know what is the mechanism. From the studies that were done in the 60s, 70s, 80s, it was hypothesized that it was the FC receptor. So there are receptors for the antibodies in the surface of the cells. And the FC receptor, the neonatal FC receptor, can be one potential mechanism for entry of the antibodies. In lupus, that was proven in some studies. In myositis, we don't know. One option in some of our patients with myositis, in necrotizing myositis in particular, is that mechanical stress may be the cause for the antibodies entering the cells. So just by using your muscles are breaking. In necrotizing myositis, we know that the muscles with the highest mechanical tension in the back of the leg are more affected than others. So in necrotizing myositis, this may be the mechanism. In other types of antibodies, it may be a different one. In dermatomyositis, we don't see the same pattern of involvement. It may be a more active mechanism of entry. And it will be different for each one of the antibodies, probably.
Gotcha. Ricky, you can unmute yourself and ask the doctor a question.
Very good. Thank you, Dr. Fernandez. A very interesting presentation. My question really is, does the environmental pathogenesis, has your research really put that to rest at this point? Are you going to just focus entirely on genetics?
Yeah, so I didn't talk about this, but I didn't talk is what causes the antibodies. So we know that in dermatomyositis, there is a higher risk of cancer. And in TIF1 gamma dermatomyositis particularly, there is an even larger risk of cancer. I'm talking about this one, but this is the only one where we have a clue where the antibodies may be coming from, because we found mutations in the tumors of patients with TIF1 gamma in the TIF1 gamma protein. So that may be the origin of the antibodies for those. But for the other, there is no association with cancer. So it may be virus or bacteria. It may be environmental triggers for those. For HMGCR, it may be the statins modifying the HMGCR protein and generating the antibodies. So I think that the environmental is going to be really important to understand where the antibodies are coming from. We focused on knowing how the antibodies were doing the damage once they were generated.
Okay, very good. Thank you.
Question. All right. Colin is next.
Good evening. Excellent presentation. I guess it's sort of, this is with single cell multiomics as RNA-seq. Is that correct?
We have done single cell and single nuclei RNA-seq, but what I showed you was bulk RNA-seq.
Okay. So I guess it gets back to what Ricky was saying. You know, and I know you've not extensively studied IBM. It's really more of the other illnesses. We've always, been told that there's some degeneration initiating event with IBM. And I guess, as we understand more and more of, these inflammatory myositis, and you start looking at genetic causes, you start looking at epigenetic modifiers, you start looking at abnormal protein, transcription and translation. I'm just wondering, if we can start moving away from this idea that IBM is completely degenerative in nature. I guess we always think about the immune system as responding to something, that there's some inciting cause that you then start getting, adaptive and innate immune responses to it. And so, this is sort of trying to modify the body's response to something that's happening, but it's not really yet what is causing that immune reaction. Am I understanding that correct?
Yes. So what I showed you about IBM, it was very little and it was not in the paper, but what we saw is that they were antibodies inside of the cells that were smaller.
Yeah. And so the issue then is, if, you know, like what Abcuro is trying to do with KLRG, you know, to start developing, you know, monoclonal antibodies that are specific for that, you know, CNM1A autoantibody, you're sort of modulating the body's response. Whatever the inciting event is, we're still unsure.
Yes, we don't know. So for the KLRG1, we know that a key part of IBM is the presence of autoreactive T cells that are cytotoxic and terminally differentiated, and this is where the KLRG1 comes on.
Yeah, because B cells and T cells, you know, are synergistic. And so if
Exactly. And I can tell you that the B cells, one of the things that we see by RNA-seq when we do RNA-seq of IBM muscle biopsies is that there is a massive production of autoantibodies locally in the muscle. Dr. Greenberg has looked into this, and they are both IgE, IgA, IgM. We have done some studies in mice, putting human muscle into mice muscle. And in those, when we take out some of the T cells, the antibody seems to go even higher than it goes normally in IBM.
Wow.
So I think that you are right. The T cells are important, but there is a crosstalk between T cells and B cells.
Exactly. And I guess, you know, to get modulation of the disease with these different immunotherapies won't really lead to a cure until we truly understand what that inciting event is, right? But
yes.
I really, really respect the work. I think it's phenomenal that we're actually moving forward in a scientific way to find out what the inciting, precipitating event is, but also, you know, how the body responds to it, and therapies, modalities that would make sense to modulate the disease.
Yeah, and if what I'm showing you is true and the antibodies are more important than what we thought, another thing that we have been doing empirically in patients with necrotizing myositis. So necrotizing myositis, 10 years ago, there were patients with really refractory types of disease that would end up in a wheelchair because we couldn't control the disease. So over the years, we have learned that by treating these patients really aggressively with combinations that sometimes entail administering simultaneously IVIG, rituxan, methotrexate, low doses of corticosteroids, in some extremely refractory cases, even adding mycophenolate, all at the same time, we can control the disease. So in IBM, this is an unknown. Nobody has combined all of these medications. Some of the individual studies with methotrexate were promising when they published, but they didn't reach significance. Some of the studies with IVIG were promising, but they didn't end up being significant. But we don't know what would happen if we combine them.
Yeah, but those therapies are non-specific, right?
Exactly. Those therapies would be targeting all the antibodies that we produce and replacing them with antibodies from other people.
Correct. And you won't be able to cure it because, you know, those stem cells of B cells, you know, if the defect is somewhere in there, then, you know, you will be able to, you know, mutate that FC chain to make the monoclonal antibody last for 6 months, but you won't cure it though. And anyway, I've talked enough, but excellent talk. Thank you.
You know, what you're saying is very interesting. I'm sure there are groups trying CAR T cells, CD19 CAR T cells in IBM. And that will probably be the most direct evidence when that is tried in IBM, if that can control the disease. We know that some patients with CD19 CAR T cells can the disease can get under control, and this is a pretty specific targeted therapy for autoantibody production. If we factor out the preconditioning therapy with cyclophosphamide, etc.
Thank you. Great question. Hey, before we get to the next question, just real quick, can you back it up and pull up the slide with the full citation?
I think it was this one. Yep.
Perfect. Thank you. If you could just leave that up for a minute.
Yeah, sure.
And Mark, you're up. Unmute yourself.
Dr. Fernandez, again, with all the others, I congratulate. That was an excellent presentation. I've studied some of this, but of course, nowhere near what you've done. I'm curious. I've had dermatomyositis for 10 years now. I have the T1F, is it Y, autoantibody.
Mhm.
Haven't cancer hasn't been detected so far. Is there a, or, I was interested in your chart. It had the predominance of the skin versus muscle, which is what I have.
Mhm.
Great skin involvement, lesser muscle, uh, and no lung ILD whatsoever. Is there a threshold you reach not having cancer for
So we
certain,
know that during
10 years?
No, we know
Like
that
me?
during the first 3 years is when the risk of cancer is increased. What we usually did for cancer screening until this year was to do a cancer screening every year for 3 years. And after that, it is considered that the risk of cancer is similar to the general population. Some of the treatments may increase it a little bit, but in general, it's similar. This year, it was an international consensus of how to do cancer screening in dermatomyositis, and they recommend only doing it at the beginning and then doing clinical monitoring afterwards. We probably will still continue doing it in TIF1 gamma for 3 years every year. And there is a new tool to kind of stratify the risk of cancer in TIF1 gamma. So recently we have described a new antibody that only appears in patients with TIF1 gamma. The antibody is called anti-SP4. And the interesting thing about this antibody is that if you have it, your risk of cancer is very low. If you don't have it, is when your risk of cancer is high. The theory, at the same time, another group at Hopkins described several other antibodies that were also present with TIF1 gamma. And the conclusion was the same, that if you have the antibodies, you have less risk of cancer. The idea would be that, the theory would be that if you have a cancer and your immune response against the cancer is very strong, your immune system can destroy the cancer. So one very exciting theory here, we have identified that some patients with TIF1 gamma, when they have cancer, the cancer has mutations in TIF1 gamma, and that may be causing the antibodies. So the idea here would be if you have a very strong immune response against the cancer, the myositis may cure you from this cancer. So now we have this extra tool called the anti-SP4 antibodies. They are still not available clinically, but we have reported them. And there are other antibodies that they have been published at the same time.
Those wouldn't show up in a myositis panel yet, right? They couldn't
They
test
are
for
still
those.
not available clinically. We have tried as hard as we could to add them to some of the clinically available panels because we think they are important because if you have this antibody, the risk of cancer is very low. And this is the main concern when we see patients with TIF1 gamma. Right. So hopefully they will be added to the myositis panel in the next few years.
Also, one quick last question. I'm involved in the Valor study. Are you familiar with that?
I'm not.
Okay. I won't ask the question then. So thank you very much. I appreciate your response.
And while we're just right off of the cancer part there, when you mentioned the 3 years, that seems to be a standard recommendation. Is that across the board for all myositis?
So we know that in dermatomyositis is where the highest risk of cancer exists. In patients with necrotizing myositis without antibodies is also a population with high risk of cancer. We know that there are groups of patients that the risk of cancer is relatively low in patients with IBM. If we are talking about classical cancer, not some types of cell abnormalities that have been described in this population, patients with SRP, patients with antisynthetase syndrome, the risk of cancer is pretty low. So here the question is if in the future, we won't need to do cancer screening in some of these patients. At this moment, we are still lying on the siding on the cautious side and doing cancer screening in most groups. But what we know for sure is that patients with type 1 gamma dermatomyositis is where we have to be more stringent with the cancer, because there is a very high risk of cancer there.
Gotcha. And do you have specific types that really pop up most commonly?
So the most common types of cancer in the general population are the most common types of cancer in dermatomyositis: adenocarcinomas, lung cancer, breast cancer, colon cancer. But in general, it has been describing most types of cancer.
Great, thank you. Dick, unmute yourself and go ahead and ask your question.
Sure. Thank you, Doctor. Are you involved with or aware of any work being done on a connection between IBM and T-cell granular lymphocyte leukemia?
Yeah, so we were looking really hard at, we have been looking really hard on that since Dr. Greenberg reported. So for those of you that are not familiarized with this, about 60% of patients with IBM, they have a type of cell abnormality. I'm not going to call it lymphoma because it's not a lymphoma in the stricter sense of the word, but it's a cell abnormality that is classified by hematologists as large granular lymphocytic leukemia. I was discussing about this particular with Dr. Greenberg, what may be the cause of these LGL cells. The most widespread theory is that they are induced by chronic antigenic stimulus. And this is where the antibodies getting into the cell may come. The other option would be that they are caused by somatic mutations. And recently here at the NIH, they have described a new disease called VEXAS, which has the same type of mechanism of disease. That is that cells in your adult body have a mutation, and that mutation causes disease. We have checked in a few patients if there were somatic mutations. We couldn't detect them, but we're still looking into that.
Thank you.
All right, and Kevin, feel free to unmute yourself and ask your question.
Thanks, Eric. Thank you, doctor. I have TIF1 gamma, I have DM, and I have TIF1 gamma, and I have ANA, IFA, and the P155, 140. Do those that are all positive and high, the ANA, IFA, the P155, 140, does that mean anything?
Yeah, so the ANA is a screening technique. So it's going to tell you if you have any type of antibody against the nuclei of the cell. If they are, like now, it is recommended that even if it is in the cytoplasm, they report the positivity, even if it is not nuclear. We know that the TIF1 gamma should be in the nuclei of the cell. In this study, we talk a little bit about that in the muscle of patients with TIF1 gamma. We also see it in the cytoplasm. But what they are telling you with a positive ANA is that you have some antibody. In your case, we know that you have the TIF1 gamma, and then the P155, 140 is the original way that we had to identify these antibodies. The 155 is the TIF1 gamma. The 140 is causing of that TIF1. So patients with TIF1 gamma also have antibodies against TIF1 alpha and beta in different proportions. So what this is telling you, probably this was done at OMRF by immune precipitation. This is telling you the same thing, that you have this type of antibodies. And
can the anti-SP4 be tested?
So the SP4 at this moment is not available clinically. But I think it will be a useful tool in the future to stratify the risk of cancer.
And I've been taking statin for a few years. Should I be not taking statin?
In dermatomyositis, we don't have any hard evidence that the statins are damaging. We usually recommend not taking statins in patients with anti-HMGCR necrotizing myositis. And in those patients, it's also important to do not take alimentary sources of statins. So there are foods that contain statins. The typical ones that we can find in the US are red yeast rice, are a type of fermented tea called puerty, and then some type of mushrooms that you can find in most grocery stores. The oyster mushrooms would be the most common ones. Shiitake mushrooms also contain statins. So I would be careful with taking mushrooms if you have HMGCR.
Okay.
But no concerns about other types of myositis.
Okay, thank you.
Thanks,
I have
Kevin.
to say, the questions are really, really good.
Well, I'm always impressed. We have some really well-studied people in the group. There's some medical professionals, and then there's other people that have done their own studies themselves and provide some really valuable information in our meetings sometimes. So, and then there's me that can barely get a sentence out. So, and that's why I just turn Zoom on, and listen to what everybody's talking about. All right, we got Dave up next for his question. Dave, unmute yourself and give us what you got.
Thank you, Doctor, for your presentation. I was just curious to know whether you're able to use AI as part of your research tools to help you ferret out the findings that you got.
Sorry, could you repeat the question?
Yes, could you use AI to help you in understanding your research results?
Yes, so we have another paper. I didn't have time to talk about this one. It was published 2 years ago, I think, actually about exploring different machine learning methods to understand these types of myositis, understand the RNA sequencing data that I was showing you. We tested different algorithms. As a computer scientist myself, this is one of the things that I have been trying really hard to do, and we identified some models that work better than others for this type of data. You had any type of artificial intelligence in mind?
Doctor, I did a poor job of introducing you here tonight. In sending out the message about this meeting tonight, I did put a lot of I did a synopsis of your background and included that out there. So I'm glad that some of your studies are included in this, as far as your resume, I should say. So it's pretty impressive. Let's see, I had one other question here. Do you have any comments on the anti-RNP autoantibody in MCTD?
Yes, so this is fascinating. So some of the studies that I was showing you from the 60s, 70s, there are some studies where they showed that the U1RNP antibodies were inside of the nuclei of the skin cells in the 70s. We are very interested in this particular one, especially because the function is related to RNA, related to RNA splicing. And we are looking really hard into this one at this moment. The problem with the U1RNP is that it's not an easy model to study. So patients with mixed connective tissue disease with U1RNP antibodies, some of them start with the U1RNP, and over the years, they acquire more antibodies. They acquire antibodies against SM. Some of the patients, they end up acquiring antibodies against double-stranded DNA. And as they acquire new antibodies, the phenotype of the disease changes. So patients that acquire the double-stranded DNA antibodies, they end up behaving more like lupus. Patients that only have the U1RNP, they have the more muscle involvement with lung involvement, a little bit of skin involvement. So we are really interested in this one, but it's hard to study because it's dynamic. It changes over time. But we are spending a lot of time. We were discussing this morning about this particular antibody. Again, really great questions.
That was from Bill Simmerall.
Thank you, Doctor.
Up to the top, we have Mike Landauer. You might recognize this character here.
Yeah, I just wanted to say another great presentation, Dr. Pinal-Hernandez. And I'm you saw some of my data. I'm an MI2 patient, and I'm also part of the NIH study. And I want to give a plug for the NIH study. And because maybe Dr. Pinal can give a little example of what you have to go through. It does a complete workup on your disease. Any questions that you have will be answered. And I'm not sure, would you do the anti-SP4 for people coming in? Would that
So
be?
we will be doing them from time to time for research purposes. As they are not available clinically, we are not going to be able to report them officially, but we will be doing them for research purposes.
So if someone came in for a clinical trial for this natural history trial, you would be able to do that SP4, right?
We will do them for research purposes, but the clinical applicability is going to be limited until it is available in a commercial lab.
And Eric's been also in the trial. Maybe you want to comment on the free housing, you know? I mean, you have to come to Bethesda, which is right outside of Washington, D.C. There's lots of airports, and they provide housing for you as well, correct? Anything else?
We treat our patients as we would like to be treated if we were patients. So
Right.
we do the best we can.
You'll get the best workup, you know, anywhere in the country.
Yeah, the good doctor missed the thrilling slideshow presentation I presented to the group a few weeks or a few months ago. I had my whole travel itinerary and everything that we went through down there. It was kind of funny. It was more meant as a joke, but I got some information out there on the study. I was very impressed. Between you, Candy, everybody down there was so professional, so welcoming. They fed me, they housed me. There was a slight bump in the road for some housing. They quickly repaired that and got me into some housing. But
I have to tell you, most of that probably was done by Julie, which is excellent at coordinating everything. So our research nurse, Julie Thompson, she's fantastic at coordinating everything.
Yep. Everything went very smooth, and even the samples taken weren't so bad. They hurt for a day or two, and it was a little bit of discomfort. But the muscle biopsy out of the thigh, that was absolutely minimal.
And all the research biopsies that we do are totally optional. So and we won't do them if we believe that they are even minimally risky.
Yep. No.
And you're taking IBM patients now, too, right? IBM people can apply.
So, could you repeat that?
Inclusive body myositis patients.
Yes, so a large proportion of the patients that we see are patients with inclusion body myositis, and we need to see as many as we can so we can move the understanding of the disease forward quickly, because it is one of the most frustrating types of patients that we see, because we cannot start the treatment at this moment. We can only recommend clinical trials, and the clinical trials are unknown.
And everything is free. Right.
It's paid by the taxes, by the taxpayer money. But
so if you can get there, it's a very valuable experience.
Yep, absolutely. I'll have to break out that slideshow maybe in March or something. We'll give people a break, but we'll break that out again and kind of go over it just to refresh people on it and hopefully get some more people down there. So it was really great meeting you down there. Doctor, I think we're all set with questions. I think the questions that we had, oh, wait a minute. Let me just see. Roy Benjamin's got his hand up.
Yes. Thank you, Doctor, for the presentation here. It's a bit over my head a lot. I'll have to go back when this slideshow comes out and learn from it a lot. But my background is in engineering and in the industry, and I spend a lot of time solving problems, and I hear a lot of solutions are assumed. It's very important to understand what the cause is before we, propose solutions. And so much I see in the medical field, a solution always seems to involve some kind of drug, etc. And I've learned today that we don't know what the cause is, and it could be environmental. It hasn't been ruled out. And I would like to see some efforts in that direction to rule that out or try to identify. And that would include a comprehensive survey of those who have, for instance, IBM, try to find some link between us. Our age, what we eat, where we grew up, what kind of water we drank, what kind of work we have done in our lives, what kind of possible exposures. And I'm not aware of anything like that. Are you aware of any
Yes, so I can tell you that here at the NIH, we are one of the groups that sees myositis patients. And as I was telling you before, we have been looking at tumors as a trigger for some types of myositis. We have been also looking for bacteria, viruses as triggers for other types of myositis. But there is another group at the NIH that is part of the epidemiology group that their main focus is studying environmental triggers for myositis. Dr. Lisa Ryder, Dr. Fred Miller, they are fantastic at their job. They have been looking really hard into environmental triggers for myositis. We know, for example, dust exposure has been proposed as a potential trigger for some types of myositis. After 9/11, some of the first responders, they had an increased rate of some autoimmune diseases, including myositis. We are also collaborating with dermatologists in other centers looking at the sun exposure. That is one known factor for as a trigger in dermatomyositis. And in IBM, it hasn't been much, you are right. But I know that they are looking hard.
All right. I think that'll wrap things up. We were supposed to end at 8. We went a little over, but that's okay. I do appreciate your time. I know everybody here is very appreciative of it as well. So thank you very much. I will be in touch just by email and follow up with you. But again, thank you from all of us tonight and from TMA for presenting. Very interesting stuff. And we're very privileged to have this most recent information.
Thank you.
I think Mark and Colin had a couple more questions.
Oh,
if you still have time, Doctor. If you got to go.
I have to say your questions are being amazing. I mean, this is really helpful for us to see what parts of of what I presented were not clear, and we have to do a better job at explaining.
Doc, do you have a few more minutes?
Yeah, sure.
Okay, all right. I saw the hands up. Those were some of the asks before. I thought they were still up from before. Let me get Howard. You had your hand up, bro. Did you have your hand up as well? Okay. All right. Colin, go ahead.
I'm really asking for other patients. In those patients who may have had the disease longer and have greater functional deficits with loss of muscle, have they looked at these abnormalities, within the stem cell of the muscle? Like, is it possible that if you can modulate the immune response that's destroying the muscle, are the stem cells able to repair the muscle? Have you done the biopsies and isolated those stem cells and looked at, is this abnormality happening within, the regenerative capacity of these cells to repair and regrow muscle?
Yeah, so the muscle is really good at repairing itself. The myoblasts, which are the cells that we were culturing, are really good at repairing muscle, but they have a limit. So if you have a disease that is not controlled for a long, long time, then you will have some damage, and this damage is usually not repairable. As part of the evaluation that we do, we usually do an MRI of the thighs. We can do it of other areas of the body. And the good thing about the muscle MRI is that it can help us separate the damage that has been caused from the activity of the disease. So one thing is that your muscles are not going to be able to grow if you train them unless there is no inflammation, unless there is muscle left. So with the MRI, we can figure out what part can be fixed with the treatment and what part is damage that has been caused by muscle being transformed into fat or fibrous tissue. But yes, the stem cells or the myoblasts that repair the muscle, they have a limit on how many times they can replicate and they can repair the muscle. After a while, you start seeing loss of function that, unfortunately, at this moment is permanent.
And so the disease you haven't really seen in those myoblasts, it's just they're exhausted over those 5, 10, 15 years of ongoing inflammation and destruction.
There are studies that suggest that they
Okay.
end up in an exhausted phenotype, that they cannot work
Yeah.
as well as they used to, and there are studies that suggest that the amount of these cells decrease over time if you have an active myositis for a long, long time. We try to prevent that moment. In IBM, we still don't have an effective treatment to prevent reaching that point. In the rest of myositis, if they are inflammatory, we usually can find a treatment to control the disease.
Excellent. Thank you.
Thank you.
Thank you, Colin. And Mark. Here.
Yeah, just a quick question, Dr. Fernandez. I participated in at NIH in the December of 2014 in Dr. Schiffenbauer's study, which is very comprehensive, lasted a week. Many tests. I don't know if that there's ever been a determination from that study or not, but it was different than the study you're head of. Is that correct?
Yeah, so before we started this study relatively recently, and it was the continuation of a study that has been going on since 1991. So I don't know exactly what protocol you were in, but it may have been this protocol from 1991. The reason why we updated the study is because it was so old that it didn't include genetics. And we thought that it was necessary at this moment to include genetics in the protocol. So to do that, we had to modify it. But we are in charge of this protocol that has been going on since 1991. If you are telling me about 2014, Dr. Schiffenbauer was in charge of that protocol at that time. So you may have been on that protocol, on the same one that we are doing now. We changed the name, but it's the same protocol.
I remember the name of it was looking and trying to determine the environmental causes of
or that maybe he's in several protocols related to myositis. It may have been a different one, but he was also seeing patients as part of the current protocol that we are seeing patients that has been going on since 1991 after the update.
Do you think it'd be valuable for me to be involved in your study?
We will be happy to see any patients that we can see, as far as we have enough time to see all of you the right way. So yeah, any of you that is interested, feel free to contact Julie, and she will be happy to enroll you in the study.
Thank you very much.
Let's see. Can you see the note there from Bill Summerall?
Yeah, Dr. Schiffenbauer and Dr. Lisa Ryder, they had this twin SIB. I wasn't involved in that particular study. Then loss of muscle and physical. So there is a comment. You should look at my case in regard to loss of muscle and physical rehab. Lost 40 pounds, left me quad, but I was able to rehab over several years to return to an active life. And I also have Dr. Mamen training in rheumatologist. Okay, so this was not a question. And I think
just a comment, because you talked about rehab and loss of muscle, and I haven't come across anybody in the last 20 years being involved who made such a dramatic recovery.
Yeah, rehab, the group that has focused more on rehabbing myositis has been the Swedish group. Dr.
Yes, I've met them all.
Yeah, and they have been fantastic at proving that rehab is good no matter what type of myositis you have, provided that you are doing the right type of rehab.
I did.
It sounds like it, Bill. All right, I think that's it. I think that's all the questions, comments. There's a ton of thank yous on there. Again, doctor, and no more questions. So thank you for your time. I appreciate it. And
thank you so much for inviting me, or for the TMA for all the support that they provide.
Thank you for the work that you do. We all appreciate it.
Bye.
All right, take care. All right. All right. We are going to wrap up. I'm just going to let you know that the next meeting, I'll send out a notice again. So our next meeting will be in February 27th, I believe. I think that's the one. Just double check here. I had it written down as the 27th. Yes. February 27th. I'll send out a notice before that, and hope you guys all found the presentation pretty good. There's a ton of information. He talks really fast. If we slow this down, it might be a 3-hour presentation. He's a fast talker.
And he
So,
has an accent from Spain,
yes.
Can you tell us when it's on the TMA site, if you hear anything?
I will. Rachel's goal is to get these on the website within 10 days of them being recorded. So she does a little bit of editing, and that's her goal, is 10 days. But as soon as I see it up there, I can send a notice out. I know people hate getting excessive emails, but at your request, I'll put that out there and let you know that it's on the website. But that was a fast and informative.
Eric, I missed that meeting with the restricted blood flow training in December. Was that one recorded?
That was not recorded. No, this was the only one that I've ever recorded, and it was more at the request of TMA, because this information could be shared not just for men, but for anybody.
Yeah.
We were lucky to get him on, with his background and the studies that he does. So, and I just happened to go down there, obviously in November, and meet him face to face, and got him to agree to come on. So we were lucky to have him. TMA wants to use that for their website, as they do some of the other Q&As. So let me see here, which reminds me.
Does anybody have any questions about the trial?